recent years, the migration of monarch butterflies has been decreasing. In 1995, butterflies occupied over 40 football fields, and this year they cover just a little bit over 1.5. According to Ernst & Cohen, the average migrating monarch flies about 80 miles per day, and it takes multiple generations to make the round trip from the monarch summer grounds to their winter grounds and back again. As studied by the University of Western Ontario, migration can be energetically expensive, so organisms must compensate in various ways, such as by minimizing excess weight or by adjusting internal organ size. Genes relating to the efficiency of a monarch's muscles are their cost to migrate. This gene, along with all other genes, are passed from parent to offspring after parent cells undergo meiosis and create gametes. Here's a brief summary of the happenings in meiosis. In interphase, the cell preps for meiosis and the cell begins to replicate its chromosomes. In prophase 1, the chromosomes condense, the nuclear envelope begins to break down, and the homologs begin to pair up. Crossing over occurs during this phase. This is where segments of non-sister chromosomes can be exchanged. The alignment of the homologous chromosomes is completed during metaphase 1. The fact that one homolog is on one side of the metaphase plate and the other is on the other side allows for independent assortment to occur when the cell divides. The homologous chromosomes are separated during anaphase 1, which leads to the daughter cells being haploid. During telophase 1 and cytokinesis, the homologs finish moving to opposite ends of the cell and the spindle apparatus disassembles. Finally, the cell splits in two haploid daughter cells. Meiosis II now begins to commence. According to an experiment done by medical students at the University of Chicago and an article published in Nature, a scientific journal, the monarch butterfly's lack of migration is a form of natural selection in regards to the gene of flight efficiency. On this specific gene, there was a mutation marked on the collagen 4 alpha 1 that created a change of haplotypes and distinguished the difference between migratory and non-migratory monarchs. The collagen 4 is an important component of membranes that help to maintain muscle function and strength. For instance, the mutation in collagen 4 can cause something similar to myopathy in humans to butterflies. In terms of genetics, the haplotypes as seen in migratory and non-migratory butterflies differed in 51 nucleotide substitutions and 15 amino acid differences. The divergent expression of collagen 4 alpha 1 and alpha 2 was what caused the difference between migratory and non-migratory butterflies and not other genes. Specifically speaking, the butterflies that were migratory downregulated the expression of collagen 4. This is where natural selection comes in. In the case of the butterflies, there is a natural selection that is acting on the efficiency of migrations and the physical demands required for long-distance flight. Changes in the flight muscles on the butterflies result in more energy-efficient consumption. However, the metabolic rates were lower in migratory butterflies than butterflies that don't migrate. The long distances required of migratory butterflies have created a natural selection for a lower flight metabolism. Overall, butterflies are reverting back to their original non-migratory state. There is a divergent selection on flight muscle function during migratory behavior shifts in order to stabilize the role of the efficiency gene. Because the monarch populations that are still migratory are very separated from those that do not, there is very little, if any, gene flow between the migrating and non-migratory populations. The separation causes each population to have an abundance of one phenotype over the other, demonstrating the disruptive selection occurring within the species. The reasoning behind these differences is strongly correlated to optimal foraging. As milkweed along the monarch's path decreases, the benefit received from traveling further decreases. Therefore, it is more optimal for the butterfly to stay in its current position as opposed to traveling farther, even though by staying it is gathering less food. This deficiency of milkweed is caused by the increase of GMO corn crops, which leads to an increase in the use of herbicides. These herbicides leak off to the milkweed crop and kill it. With this new change in the monarch's food supply, there may be an increase in non-migratory monarchs since they may have a higher fitness than migrating monarch butterflies.